Turn for Matthew Rosenberg at the Department of Medicinal Chemistry here at Uppsala University that is going to talk to us about new potential treatments or novel antibiotics. Maybe not so novel because they come from another place. We'll see. For the treatment of Clostridioides difficile. I think they changed the name from Clostridium to Clostridioides some time ago. C. difficile for the ones that don't want to deal with the name. It stayed the same. Thank you so much, Matthew, for being here. Present to us your progress and how is it going with those novel antibiotics. Thank you.
All right, thank you, Eva. Give me a second to get my screen shared. Go like this.
Yes, we see it, full screen.
You see full screen, great, and I see presenter view, so we are good to go. One moment. Okay, welcome everybody. As Eva said, my name's Matthew from the Department of Medicinal Chemistry, and today I'll be talking about one of the projects that I've been working on during my PhD, looking at, novel antibiotics against C. difficile. And I'll tell you a little bit about the story of how this project came to be and where we're at with things now. So I'm sure a lot of this is familiar to most of you, but antibiotics are kind of important. They really are a pillar of modern medicine. And that's not just for their use in treating infections, but they also enable a lot of other parts of medicine, like surgery, cancer, chemotherapy, to be done safely. And all of this kind of started in the 1940s, where the success of penicillin during and after the Second World War heralded in this golden age of antibiotic discovery. And a large majority of the classes that we still use today came from this period of time, 80 years ago. But unfortunately, as is the case with every single class, the resistance to these classes is never far behind the widespread use of the antibiotic. And in some cases, this interval has been less than 2 years since the antibiotic hitting the market and resistance being identified. And of course, for some antibiotics, because they're derived from natural products, the bacteria in the environment are already familiar with these types of antibiotics. And so the resistance may have already existed before we even knew about the antibiotic. But even with purely synthetic antibiotic classes, we still see the same trend. And that trend is rapidly becoming worse and worse. In the 2022 Lancet paper describing the global burden of AMR in 2019, which is already 6 years ago now, there were nearly 5 million deaths associated with resistant bacteria. And of these, nearly 1.3 million were directly responsible. And in their follow-up paper 2 years later, they predict that by 2050, this will have increased to 8.2 million associated deaths and 1.9 that are directly responsible. So we really do have this desperate need for new antibiotics hitting the market so we can begin to combat this growing problem. But as Long mentioned in his presentation as well, this hasn't really been happening. You can see again the golden age from the 40s to the 60s. But from about 1990 onwards, we have this discovery void where we haven't had any new antibiotics brought to the market, or rather, we haven't had any new classes of antibiotics. And the ones that have come up have been sort of Me Too antibiotics that are based on older antibiotics, but with new mechanisms of entry, for example. So why do we have this discovery void? Why is it that we don't have the drugs that we need? Well, there are a number of different challenges along the road to developing a new antibiotic. First and foremost is the antibiotic resistance itself. And we also have the issue of when doing the drug discovery, we have discrepancies between target-based activity, so affinity for the target in a bacteria, versus actual antibiotic activity in a phenotypic assay. And because of a lot of issues with the bacterial wall and being able to cross this, gram-negative bacteria are the worst case of this with all of the efflux pumps. It's extremely difficult sometimes to translate good activity in a lab to actual efficacy at treating a resistant infection. And we have a lot of economic challenges as well. The same with any drug development. There are very high costs associated, and it takes a very, very long time. And there are high chances of failure, so it's a very risky process. But something that is unique to antibiotics is we have sort of this broken market or a broken economic model where the very, very low profitability of antibiotics, we don't really want to use them if we don't have to. And when we do use them, we use as little as possible, means that it's very, very difficult for large pharma companies to profit from developing and selling these drugs, which means that there's right now basically no interest from them to actually do this. And that's resulted in this sort of discovery void that we see. And right now, the largest part of antibiotic research and development that's happening is happening in startups and SMEs and in academic groups like ours. And of course, there's no one solution to this problem. And each bacteria presents its unique obstacles and challenges. But yeah, overall, the combination of these effects means that we have quite low innovation, one might say, in the drug pipeline. Right, so that's my segue into talking about C. difficile, which is one of the bacteria that we're focusing on in our lab group. C. difficile is a gram-positive, anaerobic, spore-forming gut bacteria. It's pathogenic, and the most interesting part of this is the spore-forming part. And what that means is that under stressful conditions, C. diff will release these highly resistant spore cells. And when these spores germinate, they release toxins into the gut, usually. And these toxins cause damage to the lining of your large intestine, which causes subsequent inflammation, diarrhea. If it's left untreated, it develops into what's called pseudomembranous colitis, which is the formation of these white-yellow plaques all through the lining of the gut, caused by the immune system trying and mostly failing to fight the infection. This is very diagnostic of a C. difficile infection. And if things get really bad, then it can progress to a condition that's called toxic megacolon, which is nowhere near as cool as it sounds. It's an inflation of the large intestine accompanied by sepsis, shock, and eventual death if it's left untreated. So a C. difficile infection can be managed sometimes, but if it gets out of control, it can be really, really bad. And one of the most troubling things about these spores is that they actually survive very easily outside of the human body. So they're resistant to antibiotics, but they're also resistant to heat, they're resistant to oxygen, they're resistant to dehydration, and they're resistant to alcohol-based disinfectants, like ones used in hospitals. And so that means in a hospital setting, these spores can be really, really dangerous because they can spread to vulnerable patients nearby and recolonize them when their immune systems are compromised, perhaps. And C. difficile infection is actually one of the most common hospital-acquired infections, particularly in North America. And one of the major risk factors for C. difficile infection is the prior use of antibiotics. And repeated antibiotic treatments to try to remove the C. difficile can often make the infection worse, not better. And to understand why that is, we need to talk a little bit about the interaction between antibiotics and your gut. Namely, that antibiotics are not very good for your gut. And they cause, particularly broad-spectrum antibiotics, cause a lot of collateral damage to your beneficial gut bacteria. And this reduces what's called your gut's colonization resistance. And colonization resistance is this idea that Well, there's about 100 trillion bacteria in your gut, and they're all in competition with one another for the limited space and the limited resources that exist there. And so when a new pathogen arrives, this makes it quite difficult for the pathogen to establish itself. Or if the pathogen is already established, this really limits the outgrowth of this pathogen. Um, so your gut does have some natural resistance. But dysbiosis, which is any disruption to the natural balance, like what is caused by antibiotic treatment, creates an opportunity for pathogenic gut bacteria, like C. difficile, to come along and exploit that weakness. And particularly with these resistant spore cells that C. difficile produces, these spores can regerminate into an empty gut and recolonize all of the free real estate. Um, so the main kind of research question is, can we avoid this in some way with selective antibiotics, ones that we might be able to use to treat C. difficile, but they won't disrupt the balance of all of your other gut microbiome. So this is kind of the story of where this project started and how we are looking for selective antibiotics. I mean, it's not very easy to create deliberately. So there was this paper published back in 2018 by a group in Germany looking at the impact of non-antibiotic drugs on your gut bacteria. And in this paper, they screened nearly 1,200 drugs against 40 different bacterial species from the human gut. And included in these 40 species was C. difficile. And what they were interested in was building a resource mapping all of these unknown antimicrobial side effects that might be caused by simply taking blood pressure medication. And if they were able to identify classes of drugs that were causing problematic levels of damage to the whole microbiome, sort of focus on effects on the entire gut. But what we were really interested in was all of the data that they generated from this study. We can use this as a form of hit identification. So we were interested, are there any of these non-antibiotic drugs that had activity against C. difficile? And then the follow-up question, are there any that have activity only against C. difficile? Because the study is already in the context of all of these other gut bacteria that we didn't want to kill. And one of the drugs that we found that we ended up continuing with for the rest of the project was called Fentiazac. Fentiazac was originally developed about 25 years ago as a non-steroidal anti-inflammatory drug. It belongs to the same class, the arylacetic acid class, the same with ibuprofen and indomethacin. It's characterized by this carboxylic acid. Now, the activity of Fentiazac in the original assay was 75% growth inhibition at 40 micromolar. So for comparison, for anybody who's not familiar with these kinds of numbers, a normal antibiotic that you might use in a hospital, you need 100% reduction in growth at maybe 8 micromolar or ideally lower. So Fentiazac could never be used as an antibiotic as it is. But we saw a lot of potential here as the beginning of a medicinal chemistry project. Can we develop the structure of Fentiazac and improve it so that the activity gets better, but we retain some degree of this selectivity? So that's what I've been working with. In order to help guide our decision-making, we really needed to know a little bit more about how does Fentiazac work? Like, what really is the new interaction that's happening between Fentiazac and C. difficile? But unfortunately, the target is completely unknown. What we do know is that all other non-steroidal anti-inflammatories tested in the original study were inactive. So that means that there's no correlation between the antibiotic activity and inhibition of the cyclooxygenase enzymes, which is the activity in humans as an anti-inflammatory. So what that means is that doing rational drug design, which in medicinal chemistry describes usually how based on the structure of the type of protein, we are making changes to a drug to sort of gain new interactions between protein residues or expand into some available space in a binding pocket. We can't really do any of that here because we don't have any information on the target. So it's very difficult to do that kind of design. And what we've done instead is a more bottom-up approach. So try a whole bunch of different things and sort of figure out what seems to be working and try to progress from there. So the aims of this project was basically to synthesize a small library of analogs of Fentizac, explore the structure-activity relationship of these, so test them against C. difficile. We're collaborating with a group in Germany to do this. And then based on the results of this activity, we use that to inform how we can build improved, hopefully still selective inhibitors against C. difficile. But of course, these project aims aren't really linear, sort of starting from Fentizac and going directly to a better Fentizac. But medicinal chemistry is really more like multiple turns of this sort of cycle where we're designing the analogs, we're synthesizing them, which as a chemist, the synthesis is what really takes up most of the time. And then we test them, and the results of the testing go back. And so we do this loop, I think, probably 5 or 6 times so far in this project. So over time, we're sort of narrowing in on what the ideal structure is. So I won't talk about the chemistry itself too much in this talk, but I thought it would be useful to discuss how we are thinking about the structure of this molecule and how we're trying to optimize this. So Fentizac in the middle has this 5-membered ring, which is a thiazole. And on a thiazole, we have the different numbered positions that we can use to describe which other groups are attached to it. So we have the thiazole core. At the 5th position, we have a carboxylic acid. And then at positions 2 and 4, we have these 2 aryl groups. So when I'm thinking about the structure-activity relationship, it's really these 4 main pieces that are the blocks that sort of in my mind I'm chopping it into. But this also helps with the synthesis as well. So the key reaction that I've been using to build a lot of these molecules is called the Hansch thiazole synthesis. And despite being at the core of the molecule, this thiazole is actually the last part that gets made as a condensation reaction between a bromoketone and a thioamide. And there were a number of different strategies that in the beginning I tried to see what would be the most efficient way of building up a library of analogs like this. And the route generally that I arrived at, I will talk you through very briefly. So the first step is to build this bromoketone that we need. And the ketone itself can be created in one step from some very simple starting materials, opening up this anhydride to get our linear chain and our carboxylic acid, which in the second step can then be brominated. So that's already 1/2 of our thiazole synthesis. And the second half is simply introducing the thioamide. And in a lot of cases, the thioamides are commercially available and not too expensive, so we can just buy them and use them directly. But in case my supervisor tells me that something is too expensive and I'm not allowed to buy it, I have to make it myself from perhaps an amide or some other cheaper starting material. So a number of the analogs needed quite a lot of adjustment to this synthesis, but kind of in a nutshell, this is the general scheme. So 3 or maybe 4 steps overall, depending on the analog, which is pretty efficient. So using that synthesis, I've made to date, I think, 40 analogs. And before I talk you through the data and kind of where we're at so far, I think it's worth taking a minute or two to discuss the actual design of these assays, because we're not just using MIC values as the metric here. We actually have growth curves. So what you can see is the growth of C. difficile in an anaerobic growth media with the analog added, and the optical density is measured every hour for 20 hours. So we get these nice curves. This is done by a robot. It's automated. There weren't any undergrads that we forced to stay in the lab for 24 hours to get this data, thankfully. And so how we're interpreting this data is we draw this line, which is the cutoff for determining the area under the curve. And this cutoff is when the control enters into the stationary phase, so where it stops the period of accelerated growth. We can then calculate the area under the curve for the normal growth, and the metric of growth inhibition is taken as a percentage reduction in this area under the curve up until the cutoff. So for the rest of the talk, you'll just see percentages, but that's what this percentage means. It's the reduction in growth area of the growth curve. Right. So starting off, we have the 4 position of Fentizac, this lower arrow ring. This was one of the first series of explorations that I did, and a lot of the changes were pretty minor, I would say. We have this chlorine that I'm moving around the ring a little, replacing with methyls, fluorines, methoxy, ethers, or simply removing the chlorine completely. And the results that I got from this were terrible. None of them worked, not even a little bit, which feels like a negative result, but it was actually really interesting for us because it means that whatever interaction is happening, it's very, very sensitive to change. It does mean that we don't understand yet what this interaction is, but because we know that if we change it at all, we completely lose activity, well, that's quite an easy decision to make. We simply won't change it.
One question before so 0% means not that is equal to the control, but that it lose completely the activity.
Exactly. So 0% would be 0% reduction in growth. So
okay, not 0% reduction in the area. That means equal to control.
0% means equal to control.
Okay. So it means that it didn't do anything, but it also didn't make it worse.
Than control? Yeah, exactly. So
Okay.
it didn't grow better than the control. I mean, it can't use these analogs as food. But
okay, but the control is nothing added. Not the control is the original drug.
yeah, sorry, yeah. The
Okay.
control is with no drug. It's purely
Okay,
the
okay,
global.
sorry. Maybe I just missed that. But okay, now we're in the same page.
No, but that's good. Important that everybody's with that because there'll be a lot of percentages coming after this. Okay, yeah, so 0% means that all of these did not inhibit the growth of C. difficile at all. Moving on to the position 2 arrow group, here we had some slightly more interesting early results. So in the left box, you can see some of the larger changes that I was making still resulted in no growth inhibition. So these were all completely inactive. But having a methyl group gave us some activity. So the 30% is still worse than the 75% inhibition that Fentizac had. So we're still at this stage not even improving on Fentizac, but we did find the potential where, okay, some changes in this area are tolerated. So expanding on that, changing some larger groups, adding in some oxygens or fluorine, we were able to get the activity to be the same as Fentizac, which is not that much of an achievement by itself. But it's very interesting that such small changes, similar to the other position, can result in the activity dropping off completely. But we were encouraged by this lowest result, the 75%, with the fluorine there. So the next round of analogs, we were thinking, okay, let's try some more halogens, similar atoms and similar positions around this ring. And there we started to see actual improvements in activity and quite a bit of a jump, where we start introducing some of these larger halogens around this ring, bromine and chlorine. And here we now have some synergistic effects. So if you look at, say, the top analog in the left box with this chlorine in the meta position, by itself, not particularly active. But if we start introducing additional chlorines or we start introducing another fluorine, we're now seeing the activity jump up even more. And so we were really encouraged by this. We're thinking, okay, well, are there other combinations of sort of two halogens that we can start to get up to this 99% inhibition mark at 40 micromolar? And the answer was yes. So it was this particular pattern uh, where just changing out one of the halogens and leaving the other the same, you can see the trend top to bottom. I mean, we've increased from 96 to 99, but overall, everything is looking pretty good. And the real trend that we observe here, you don't see until we look at the activity at lower concentrations as well. So we're starting to actually get an activity curve. So these are the same analogs again as we had in the right-hand box. And now we can see that at 20 and then also at 10 micromolar, we are, in some of the best analogs, still getting pretty good inhibition. So this one at the bottom, 72% at 10 micromolar, is very comparable activity to Fentizac, but at a quarter of the concentration. So this was really exciting. Yeah, so moving on then, we have changes that we made to the thiazole core itself and also to this carboxylic acid group. The initial changes to the core were quite large, sort of swapping out some atoms in the middle, removing an entire group, or this molecule on the right is all the same groups but swapped positions with one another. But none of these had any activity at all, which is not so surprising because these are quite drastic changes to the whole scaffold. So we can say, okay, the thiazole core and the general scaffold, we do need to keep this. But looking at some changes to the carboxylic acid, we can reduce it to an alcohol, remove it completely, or swap it out for an amide, which in a lot of cases is a very, very similar group. None of these had any activity either, though. So we can conclude that, okay, well, we definitely do need the carboxylic acid at this position as well. But one other thing we tried was that instead of changing the acid itself, we can simply change the length of this carbon chain that it's attached to. And if we truncate it, just removing the one carbon in the middle, activity completely dies off. But adding in one carbon actually leads to quite a marked jump in activity. So just to compare, I guess, we're seeing 96% at 40 micromolar and 92 at 20 micromolar. So if I go back a couple of slides, that's already comparable to some of the best of these dihalogen analogs, but it still only has that original group. So that was also quite an exciting discovery. And what you're sort of thinking is, okay, well, what is the picture then, right? So to summarize, this is kind of everything that we have learned in one picture about the structure-activity relationship here. So the position for our group down the bottom is essential. We can't change that. The core itself as well can't really be changed, but extending the length of this carboxylic acid chain improves the activity a lot, as does having this dihalogen pattern on the position 2 arrow ring. So probably definitely the obvious next step, we're looking for synergy. So changes at one position created quite a big jump in activity. Creators at a second position, similarly large jump. What happens if we put them together? Unfortunately, I don't have the data for this yet. These compounds have been sent away just a week ago. So hopefully sometime soon we'll be finding out about these. But I am very excited that we'll see an even larger improvement in activity based on some hopeful synergy between these two changes. And lastly, the next steps for the microbiology, first of all, will be to rescreen the best analogs against the original panel of gut bacteria. Because the objective this whole time has been, yes, improve the activity, but we still need to have this selectivity in the context of the human gut. Otherwise, we're not going to be as effective in actually treating C. difficile infections. Um, and we also want to test it against some clinical isolates of C. difficile, as well as some whole gut microbiome isolates that have been infected with C. difficile. So basically, will some of these analogs be able to reduce or remove the growth of C. difficile such that the other gut bacteria can grow uninhibited? So that will be very interesting. And the last thing that we're looking into is whether or not any of these analogs still have their anti-inflammatory activity. Because C. difficile infection causes inflammation of the gut, right? So we were thinking, okay, if any of these drugs also have good anti-inflammatory activity, or even some of their original anti-inflammatory activity, that may provide a second useful activity to add on top of the antimicrobial activity. Um, so we're also waiting on results of this testing now. So I don't have anything to share today, but we are very excited to see if we have the possibility of sort of a dual action molecule. So with that, I'd like to say thank you to the research group at Uppsala University, our collaborators in Tübingen and Stockholm at KI, and of course, thank you to UAC for the funding.
Thank you so much. You're getting some applauses. Now actually they show up in the screen. That's nice.
Hmm.
Great, great, Matthew. Very, very cool work. Let's see if someone has questions. Again, you can put your hand up. Oh, Michael, Michael, Mikael, you have a question right away. So go ahead.
Hi there. Can you hear me?
Yes.
Yeah, I can hear you.
So, Matt, exceptionally clear, beautiful talk. Congrats.
Thank you.
Yeah, so there are several different questions, but one that really tinkled my curiosity was this. I remember reading a paper 2, 3 years back about repurposing of gram-positive drugs for gram-negatives and the do's and the don'ts. And one thing that I remember from that was that a carboxylic acid addition is typically a bad idea if you want to target gram-negatives. And here you see that this carboxylic acid is also vital for your activity, but the length of it is really key to that. Are you discussing this sort of pairing these functional tests between C. diff and let's say E. coli or some gram-negative to see what it does to selectivity? Because if that paper is correct, then that should also matter a lot for the selectivity. Gram-positive versus gram-negative, I mean.
Yeah, exactly. I mean, in the name of selectivity, if we would expect that having a group like this means that it's bad against gram negatives, but stays good against gram positives, then that would be beneficial, of course. We have not tested against E. coli, but I mean, we probably should.
I mean, you will, of course, know in your final screen against the whole panel, but
yes,
I mean, in this testing, it seemed from that to be a sort of a good idea to have that pair of a gram-negative and C. diff.
Yeah, that's a really good suggestion, actually.
Then I have, from a biologist point of view, maybe a bit of a naive question, but that will come up in all these synthetic chemistry approaches coupled to a functional readout. Many of the reagents one uses here to generate the different modifications are by themselves, they can potentially be very toxic. So how is purity controlled for it? I mean, of course, you do some chromatography or NMR to say, yes, there is the clean substance is majority. But is there some sort of cut-off reasoning to say, yes, the impurities across all your samples you're generating is always below this certain level, and therefore, if you have a hyper effect, it cannot be a contaminant? How do you reason around this? Just for educational purposes, I'd like to know.
Sure. I mean, I think typically, and this is not just for my work, but this is just in general. 95% purity is considered an acceptable cutoff. But in terms of the analytical data that we look at for when we're preparing these compounds, right after purification, we'll typically use NMR spectroscopy to analyze the structure of the molecule itself, and then also some kind of LCMS or HPLC to determine the purity by UV absorbance and TIC. And I would say, like from the perspective of a chemist, a compound that is 95% pure still looks really bad. And most chemists will try for much, much better than that. But it's a valid concern, of course. Like it's impossible to completely eliminate every possible contaminant. And if there are questions about the purity of any one sample, probably the solution is simply to resynthesize it or retest it, repurify it. But if the synthesis and the purification would be done in the same way the second time, that's probably difficult to control. If there would be some very, very, very small amount of something that was very, very potent.
I guess you could probably predict some of the precursor reagents one could predict to be very toxic or not. So I guess that's
yeah. I mean,
indications.
there are certainly known reagents, known byproducts from types of chemistry that are very important to get rid of. Like lots of chemistry is catalyzed by copper or palladium or other kinds of heavy metals, which obviously you don't want any of those in there. But there are many other sort of reactive byproducts that can interact with protein residues or DNA in an assay. I mean, there aren't any that I know of that could be byproducts from the chemistry that I've used in these projects, but I wouldn't bet my life on that. Like there always could be something that I don't know about that's in there.
Right.
Great.
Yeah, well, thanks a lot from my side.
Thank you, Mika. Thank you for the question. I was actually thinking regarding, now you started not really knowing what the target of your first drug is within C. difficile, and then you started doing changes. Has that given you more insights or some ideas into how this might be actually working for C. difficile? Or are you going to take this forward to try to find the mechanism of action? What are your thoughts and progress on that? Or is it needed at all? I don't know.
Well, I mean, that's kind of the thing, is that earlier on in the project, I was really stumped and very frustrated by a lot of these kind of early negative results, thinking, my God, if only I knew more about what is actually happening, then I could make more educated guesses into what types of changes that I need to make. But I guess as evidenced by the fact that we have been able to improve the activity quite a lot, even while knowing nothing about the target. You don't need this information. Eventually in the future, I think if this project would be kind of continued after I finished my PhD, then there would reach a point where, okay, we need to have this kind of information to make other types of optimizations. There are
also,
only
even if you to see if you unravel maybe some essential mechanism within C. difficile that it wasn't really known before, right?
Yep, absolutely. I mean, we have talked with our collaborators in Tübingen about this, and the answer is essentially, if you have some molecules that are really potent, then we can absolutely look into it. But that's somebody's PhD project. It's way too unrealistic to have a chemist do that kind of a work, or to ask somebody to take on a side project.
Yeah, I was just because I was thinking, in the recent years, there's been some publications about how can you, in a sense, use large language models and neural networks that have been trained in a lot of different molecules that know what they are actually targeting to, and then do somehow, oh, this might look like this, it might actually be that, and then you have a starting point to test in a sense, if that could be a more directed approach, or even possible of just one afternoon, oh, I'm gonna put these molecules through these kind of systems, if that's available to give us, I don't know, a wild guess, if anything, but I guess nothing of that has been done yet.
No, I mean, not in a scientifically rigorous way.
Yeah.
I have tried a few of these types of models myself, and the output that I get usually is, these look like they might be quite good at inhibiting cyclooxygenase 1 and 2. And I'm like, I know. I know, they're anti-inflammatories, yes. But I think because C. difficile is much harder to work with than something like E. coli, there's pretty limited data out there for these types of models to use.
Yeah, you basically are only looking into what's already there, and if there's not so much, then
yeah, exactly.
Yeah.
But I mean, I am extremely interested to find that out. But
the biologist in me is like, I want to know why it doesn't work. Why is it like, you know?
Yeah. You might be waiting a while.
Yeah, well, we can wait. Great. Thank you so much, Matthew. Any other questions? Any other one here wondering about your beautiful chemistry? There's so many. How long did it take you to make all those analogs?
That's a good question. I mean, this project has been like not continuously, but sort of start to finish. I mean, this has been my whole PhD, I guess. But I mean, I've been working on other projects. It's not like this took 5 years.
Yeah.
A round of 10 analogs could take 3-4 months, something like that, depending on how much new chemistry I need to investigate to actually build these.
Yeah, quite a lot of time.
Yeah, it can vary a lot.
Mikael, you have one more last question?
Yeah, so there was a minute left or so that I can just briefly chip in one more. So are you also considering here for selectivity the tox on human cells? Because obviously you want this to be administered in the gut. And like these dual chlorides that you saw were really potent. They may be potent at causing headache for human cells as well. Is that included in the pipeline downstream?
Yeah, we already have some tox data on I think it's Hep G, like Hep G2 cell tox. And there's no toxicity there. Obviously, if we want a more accurate toxicity readout, doing some sort of an assay with like CACO2 cells would be more appropriate, like, intestinal epithelial cells. Something else that is, I think, worth thinking about along those same lines is that we actually, in contrast to what you normally want from a drug that you might take orally, we don't want these compounds to be able to cross over the gut membrane. Like if they're able to just accumulate in the gut and not really be taken up by humans, that's an ideal scenario. So in optimizing for that, we might kind of avoid potential toxicity effects anyway, because, well, yeah, it's not really going into your body in the same way. But we don't have any assays designed to test or measure that. From what I understand, it's quite difficult to set up.
Well, our lab does, but
I was gonna say, Mikael, maybe you can.
Yeah, we can have a coffee at some point, and then I'm sure Shai is here as well. She knows a lot about the general tox screening, but when it comes to intestinal specific models, this is exactly what we do. But okay.
Oh, well, that's good to know.
Subject for later.
I'll be in touch.
Lovely. Yeah, I was thinking because, with your organoids models, which is actually epithelial intestine, right, and gut, you potentially could put the drug there and see actually how the cells react to that, right? That's cool.
Thanks a lot, Matt.
All right. Thank you, Mika.
Great. Lovely. I love to see these things happening and coming across paths. Very, very cool. With that, if nobody else has any questions, we have reached the end of our webinar. This September of 2025, first event for this autumn semester. And we'll be following up with more webinars this semester. We are also having our UAC 2.0 kickoff in 2 weeks with our members as well, and preparing for Uppsala Antibiotic Days 2026 already, which will be happening in June 2026. So keep your eyes up, check our website, our news channels, and I hope to see you at one of our next events. Thank you, everybody. Thank you, Matthew, for your presentation. Bye.